My name is Stephen Benjamins. I'm a marine ecologist working at the Scottish Association for Marine Science up here in Oban. And I've been asked by Jane to give you a brief overview of some work that we've just started doing on quite an interesting project on photo ID. See if we can use that as a tool to identify individual skates and to see what we can use that for managing these species. This has already been covered to some extent by Jane and James, of course. Why are we interested in these common skates? The main reason being because they are no longer as common as they used to be, primarily due to the fact that they get caught in, historically in commercial fisheries and nowadays mostly as bycatch. These are large, long-lived species with a very low reproductive rate and as such quite vulnerable to that kind of mortality, which means that the species is now absent, effectively, certainly ecologically absent, from most of its historical range. There are a few little remnant populations that we are currently aware of, including the one just out here. In order to manage the species effectively, and we have heard about several different management options, it's important to know the population size and how individual animals use the area that we've protected. So their distribution. So how do we, an important thing about that is you need to tell different individuals apart. You need to know what each individual fish is doing if the population is as low as we suspect it might be at a global level. Now, just to clarify, I am not a skate biologist. By training, I am a marine mammal scientist. And in marine mammal science, we don't tend to, well, there is an element of tagging, but there is also the use of a technique called photo identification. So the use of photographs taken by scientists or members of the public you can scrutinize those for individual features and work out that that picture is of the same individual. So this is just a snapshot of two individuals from the bottlenose dolphin population in the Moray Firth, which is managed by our colleagues at the University of Aberdeen. Bottlenose dolphins have very distinctive along their dorsal, indicated by by the red arrows, some nicks along the trailing edges of that individual there, and some rake marks the teeth of another dolphin have left behind. And the animal on the right, similarly, there's a very clear, lightly pigmented line along the edge of the dorsal fin, which translates from the left and from the right side. So that particular population consists of several hundred individuals that have all been photographed like this. And it turns out that these marks, in bottlenose dolphins at least, are quite stable. So you can come back the next year, take another picture, and you can use that to match to your existing catalogue. And that indicates that that dolphin, at least, is traveling back and forth to that area. And we were curious to see whether this might work with skates as well. Now, just a quick word about photo ID as a methodology. It's complementary to tagging. It's a non-invasive method. Anybody with a camera can do it. But it does require that certain assumptions are met, and I'd like to discuss those briefly with you. First of all, it assumes that the individuals in the population that you're looking at can be, in fact, reliably distinguished through visible marks. Now, this is a picture from an elephant photo ID project where it's actually the lower outline of the ears that turns out to be uniquely identifiable and stable throughout the ears. That's the second point. The marks need to be stable over long enough periods that that it's relevant for management. It's no good if the animal changes its appearance on a regular basis, which is why photo ID in birds is actually not that commonly used, because they shed their plumage so frequently. The third point is to do with the number of individuals that you can actually match. The proportion of identifiable individuals in a population needs to be relatively high. So it's no good if only 1 in 100 individuals is actually uniquely identifiable by eye, and the rest of them all look the same to us. That becomes completely useless. The fourth point is that it only works in animals that don't occur in millions. You need to have a population that is moderately sized, up to a few thousand at most, shall we say, for ease of counting. And finally, it needs to be practical. Individuals need to be detectable, or you need to be able to photograph them without going through excessive effort, shall we say, the BBC Nature Unit's photography approach. We'd like to do it a little bit easier. So skates, as it turns out, are quite suitable for photo ID in that they are large, they have clearly identifiable marks that I hope you can see here differ from individual to individual. We don't think there's that many of them out here in the MPA, for instance, and they seem to be at least partially resident. So this could be quite a useful way to keep track of where these animals are going. How do we, in actual fact, go about doing this? In practice, it turns out that you spend a lot of time comparing one photograph to another. 2 computer screens, and away you go. And I spent most of last year's Christmas holiday doing this. We tend to use, let's see how this works, the whites, the pale spots, and it's the spot patterns that differ. And I've tried to highlight that in this manner. And just have a look, for instance, the spot patterns on the nose of these different skates. You need to get your eye in a bit, but those patterns differ. The spot patterns on the trailing edges of the wings differ. And once you start looking at a lot of these, you start to realize that, yes, they are in fact all different. And as far as we can tell, those spot patterns remain stable. And we've tested this by comparing the spot patterns to the fish that James tagged, and that we therefore know must be the same individual. And as far as we can currently tell, those spot patterns remain stable. We also, of course, just for confirmation, use other marks, like nicks in the fins, for instance, in the trailing edge of the wings. But some of those, like scratch marks or rake marks, are non-permanent. And that in itself is another interesting scientific question. How fast do those marks heal? But for the present purposes, we'll leave that to one side. But that is something else that we're interested in. We see this as very much complementary to the work that James and Jane are doing in terms of pit tagging. It does allow confirmation of the identity of a fish in cases where there are no pit tag scanners available, for instance. It also allows you to incorporate information directly from members of the public, other anglers from other boats, because a lot of this work is based around the data provided by the skippers locally. If people have historical pictures before the pit tagging started, that allows you to confirm that that fish was actually resident in, or not, for a much longer period in time than we would otherwise know. And by looking at these photographs, they can also inform you about other things than just their animal's identity. It can tell you about its general health, the presence of dermal skin diseases, for instance. We've only scratched the surface, really, at this point. So, what do we have? When we started with this project in November last year, we started with just under 500 photographs, which were provided to me by Ronnie. Thanks very much for that, which spread from 2011 to 2016. And then subsequent to that, I obtained another 170 odd photographs from Jane and James, obtained during their tagging exercises. Which, if you combine all that, it comes to about 413 capture events, which I would define as an individual skate being landed on the boat at any one time, over a total of 165 days worth of fishing effort. As we've heard before, this is no different to what James found. Most of those are females, as illustrated here. You'll see that the bulk of the data is actually concentrated in these last 3 years. 2016 was a particularly good year, so there's a lot of individual capture events in this graph. So each event represents an individual skate, but some of those may have been recaptured multiple times. Most of the pictures, again, no surprise, the vast majority of these were females, and the vast majority of the pictures were of sufficiently high quality that we could actually start using them for a matching effort. I should also highlight that the 2017 data are currently being processed, but I don't yet have those results in here. We're just catching up with the backlog. So a brief summary of what we do have. At the moment, our photo ID catalog for the MPA, based on Ronnie's photographs and SNH's photographs, results in a catalog consisting of 251 individuals. Of those, 34%, so approximately 80 or so, have been recaptured at least once during the time period covered by this photo archive, which goes back to 2011. That recapture interval varies fairly dramatically. Some fish seem to be very keen on the bait that is being put out. I should also point out I am not very knowledgeable about skate angling, but it does surprise me a bit that the skate that had just gone over the side, within an hour, it is back on deck. I wasn't expecting that. But on the other end of the scale, the longest intercapture interval was 1,600 odd days, which is just under 4.5 years, which I thought was quite impressive, which is about the longest extent possible in the database as we currently have it. So we are currently engaged in a bit of an exercise to collect more pictures from anybody who might have them. Primarily, if you have pictures from further back, if you were fishing here 5 years ago and you took some photographs, we would like to know if one of these 251 fish is in those photographs, just to confirm that it was already there way back then. Alternatively, if you go fishing somewhere else, outside the MPA, and you have some photographs, it would be really interesting to know that some of the fishes that James has tagged, for example, turn up elsewhere, as it were. So, if you are now inspired to take more photographs of any skates that you land, we would just like to point out some tips to make it slightly easier for ourselves for the matching process. The picture on the right represents pretty much an ideal photograph of a skate. So here you see the fish is laying flat on deck. The spot pattern is actually quite clear, it's clearly visible. The photograph is taken from above, so you see everything in one plane, as it were. The picture is in focus. The whole body is visible, including, in this case, the claspers of the male, so you can clearly identify it as a male. And there are no reflections or shadows, weird tricks of the light that makes it more difficult to pick out the spots relative to any effects of light. If you contrast that with the picture on the left, that is pretty much the worst kind of picture you can get. It's very difficult to do much with. This happens to also be a relatively poorly marked individual, which doesn't help. But you'll see, on the one hand, the whole right wing is in shadow, and most of the left wing is covered in reflected light, which is unhelpful. So that's just a brief summary of where we are at the moment. We wish to move this forward. We are currently busy matching the 2017 fish, including those from other people. We are going to, something that James and I need to discuss, but we need to try and estimate abundance using both the tag data and the photo ID data to see if we roughly come up with the same sort of numbers, which would be helpful. And we then need to integrate the databases that are being generated by SNH on the basis of pit tagging and my photo ID database, which is currently being held at SAMS, so that you have essentially a picture that matches a pit tag. And at the moment, that doesn't quite exist yet, but we're working on it. I would also like to mention that we have potentially some other ways to monitor skates. This summer, Clive Fox came last and I set out with some preliminary lander work where we had a baited camera lander with a GoPro essentially sitting on the seabed. We deployed that 5 times for a period of 2 hours each. They were very brief deployments just to see if it would work out here in the central Firth of Lorne between Carra and Mull, more or less. And what you see is essentially what you get. This was just to see whether it would work, whether we could attract skates to a baited lander. So the lander was not designed to attract skates, it was designed for another purpose. So it essentially consisted of a rectangular base with a pyramid structure with a camera in the middle looking down, and we strapped a mesh bag in the middle with some octopus and mackerel in it. And at least 7 individual events of skate visits were recorded during those periods, including 4 which were obligingly enough to actually sit still underneath the camera for a bit so that you could clearly see the spot pattern. And those 4 fish were all different. And we then matched them to the catalogue and found that none of them were in the catalogue at that point, which was in itself interesting, given that what we just saw in terms of James's data, how localized some of these fish are. That's perhaps not surprising, but it's good to see. So where do we take this from here? We are going to hopefully continue to update the skate photo ID catalog as more and more pictures come in. We are currently in the process of trying to automate the actual matching process, which is the most tedious part of the whole procedure. If we can automate that at least in part, that would be helpful. One of our students is currently working on an assessment of how permanent the spots and the different marks actually are. I should have mentioned we spend a lot of time looking at the pale spots. Skates also have dark spots or black spots. We don't know how they are formed or whether they are permanent or not, but that is an aspect that we also want to look at. As part of this exercise, SNH is providing financial assistance to us, and we will therefore be providing annual reports on how the cataloging work is going to be going. Later on, as I mentioned, we wish to estimate absolute abundance to have some idea of how many skates might actually be out there. And we would very much like to continue or expand upon some of that lander work, including putting landers in where most of the anglers don't go, because that's essentially a gap in our coverage at the moment. So that's most of what I really want to say. I just want to thank quite a few people who helped out, including first and foremost our skippers, who have been incredibly helpful. This project would not exist without their help, as well as our students, who have been very heavily involved in a lot of their volunteering their time in a lot of the data collection, and of course SNH for their financial support for this.